Hello, my name is Herbert Sauro. I'm from the University of Washington in Seattle in the United States. I want to thank the organizers for inviting me to this conference. I'm very, very sorry that I'm not able to attend in person. I had some problems with the visa application. It was refused. So I think the best we can do at the moment is to present the talk as a video, but then I'll be on Skype afterwards, where you can ask questions and so on. Okay. So the first thing I want to talk about is some of the work that we do in my lab. I guess my main interest ever since I was an undergraduate has been how biological cells work. They're beautifully complicated things, and I've been very interested in trying to understand the operating principles of cells, especially how the systems are controlled and regulated. And in that vein, I've worked over a number of years on many different topics, but currently these are our three main areas. So the first is experimental synthetic biology, which I'll mention a bit later on. But here we've got two experimental programs. One is related to how noise propagates through networks. As you may know, there's a lot of stochasticity in cells as a result of random reaction events. And when the number of molecules is small, we tend to see a lot of noise propagating through the networks. And we currently have very little theory to describe that. And experimentally, there hasn't not much has been done in this area. So the last 5 years we've developed a lot of theory, and we've now been doing some experiments on noise propagation. Some of this has been published, and we have more work that's going to be published soon. The other experimental area is evolutionary stability of synthetic networks. One of the things you'll find is if you do modify an organism, you'll find that there's selection pressure against your modification. The reason for this is that often the modification results in a metabolic load, and that tends to slow the growth down a little bit. And if there are any mutations that knock out your circuit, of course the growth rate goes up, and there's selective pressure for the wild type. So one of the things we're interested in is how can you slow down that? How can you make synthetic networks more stable to evolutionary selection? And so we've been developing techniques that modify the DNA sequence, and now this year we're starting on a new program to develop active circuits that try to resist or try to reduce the impact of mutations in synthetic networks. On the computational side, because I've been interested in simulation and modeling for a long time, and more recently I've been interested in how we actually do models in biology. I mean, it seems to me that we've been taking the methods developed in the 19th and 20th century that really apply to simple models, and we're applying them to cellular models, which tend to be very complex and also tend to be sparse when it comes to data. And so one thing I'd like to do is to create some very high performance simulation tools. One of these is called libRoadrunner. This will basically create a model, it'll convert a model, a differential equation model, into machine code effectively, and it'll run a model at full speed on a desktop. And there are groups around the world that are using this on large clusters to simulate extremely large simulations of the order of hundreds of millions of simulations at a time. And the idea here is that you basically generate an ensemble of models. So rather than a single model, you generate an ensemble, and from the ensemble you get an idea of how the system is going to behave. And it's the analogy is with the way the weather forecasters do weather forecasting today. They don't have a single model, but they have multiple models which they run, and from the ensemble they gain an idea of what the system is going to do. The other thing we do, of course, we create user platforms for systems biology and synthetic biology. The most recent one is a Python platform we have. Integrates many tools together, including libRoadrunner, a number of other tools. This seems to be very popular. The idea is to give a MATLAB experience but from the point of view of Python. So all our software, of course, is open source and freely available on GitHub and SourceForge. The other tool we have, this is the one I personally maintain, is a visual tool called Pathway Designer. This allows people to draw their networks on the screen and then do various simulations and so on. The other big area I've been involved in the last 15 years is standards, and now more recently reproducibility. In the standards domain, I started back in 2000 developing the systems biology markup language. This was in collaboration with Mike Hucker and Kitano and Hamid Balori and Andrew Finney. And Mike in particular has been working on this since, and I've been assisting somewhat. So this is a standard that allows you to represent models, so computational models of cellular systems in a machine-independent way, so that if you produce a model, you can give it to somebody else in this format, and then they can read it in. One of the problems in computational systems biology is reproducibility, and it turns out that most models, for the vast majority of simulation models that are published in the literature, are not reproducible at all. And the reason for this is that often there are errors in writing up the model. Either units are wrong, numbers are wrong, the model itself is wrong. So the idea here is to use an electronic format instead, which you save directly from your simulation program, and so there's no ambiguity then in publishing the model. The model you publish is the one that you actually ran. And to assist in that, there's also a standard called SetML that's been developed primarily in Germany, but we've been involved in it too. This is SetML. This is a simulation description language. What this does, this allows you to describe how you wish to do the simulation. So SBML describes the biological components of the simulation, and the SetML describes what you want to actually do with the simulation. And finally, another standard that I initiated was SBOL. This is the Synthetic Biology Open Language, and this is going to be the main topic of this talk. Okay, now we're moving into a new era, technological era, that's centered around biology. But if you look back in history, you'll see that technological leaps have had major impacts on our development, and we can start, for example, with the discovery of bronze and how that changed warfare. We can move on to the Romans, who invented concrete and had a significant impact on their architecture and the ability to manipulate water and so on. And then more recently, we had the development of the steam engine, which heralded the Industrial Revolution. And now more recently, in the last 60 years, we have silicon. And that's, of course, revolutionized how we communicate and how we deal with information. So some would argue that the next technology that's going to drive things is biology. How, when, it's uncertain, but it's certainly moving in that direction. And of course, one of the key areas that's been developed in recent years is synthetic biology. This is to be distinguished from genetic engineering, which tends to be more simplistic. Genetic engineering tends to focus on one gene at a time, whereas synthetic biology will manipulate entire networks and gene operons. So just to make sure we're on the same page, what is synthetic biology? So at the top of this slide here, let me just get the pointer. This is a quote from the Royal Society. Synthetic biology is the design and construction of novel artificial biological pathways, organisms or devices, or the redesign of existing natural biological systems. So it's essentially treating biology as a technological substrate, where we can manipulate the technology either by adding to it or taking away from it to produce some desired outcome. And there are potentially many applications, of course, to using biology. It's a unique technology in many respects. I mean, it's a wet technology. It's not a solid state like silicon or concrete or bronze. It's wet gel-like technology. The other problem is, of course, this technology evolves, which is a problem in its own right. And of course, the technology can reproduce, which is pretty unique. But there are many possible applications, from new materials to sensors to new structures or new ways to process information. And of course, there's the more traditional applications of metabolic engineering, where we can, use organisms to create drugs, etc., etc. So this is what synthetic biology is. It's been going now for some years. I guess this slide is a bit old. It's now older than 12 years. But back in 2000, two landmark papers were published. One was the construction of a genetic toggle switch, and the other was the construction of an oscillator, now called the repressolator. This is the first time, I believe, that somebody had engineered a set of genes to create new behavior. Prior to this, most genetic manipulation involved single genes, and the amount of novelty that was introduced was fairly minimal. But here we have completely new behaviors emerging from this re-engineering. And so since that time, synthetic biology has become more and more popular. It's interesting that it's really been attractive to engineers rather than biologists. Many of the best synthetic biologists are engineers by training rather than biologists, and I think that suggests the direction in which synthetic biology will go in the future. So I started in synthetic biology back in 2007, 2006, when we published a paper on 3 what are called feed-forward networks that we created in E. coli that acted as range detectors, so band detectors. And after that, I started to realize that, well, how would somebody reproduce what we did? I mean, what information do they need? And it turned out there's a lot of information you need to reproduce a synthetic design. Not just the DNA sequence, but of course you need the organism, the strain, how was the DNA sequence packaged, what growth conditions were used, and so on and so on. And so I started thinking, well, maybe we need some kind of standard way of representing synthetic biology designs. And in 2007, 2008, I got a grant from Microsoft to kickstart the creation of a community, development of a community that would start thinking about how we could standardize designs in synthetic biology. And a couple of years later, we published this paper. Well, it was a letter to Nature Biotechnology, where we discuss the issues in reproducibility in synthetic biology. I mean, to be honest, if you take any paper in synthetic biology, I suspect 99% of the time you won't be able to recreate what they did. And that again is due to just errors in writing up the paper. Sometimes, for example, people won't give the entire DNA sequence, they'll just give you the parts list, but it turns out that the intervening spaces between the parts is just as important as the parts themselves. Or they may miss out on some specific growth conditions that you used, or they missed out on when the measurement should be taken in log phase, or stationary phase. Or was it batch, or was it continuous? How was the DNA inserted into the organism? Was it in a plasmid? What plasmid? Where in the plasmid? Or was it inserted into the main genome, and where on the main genome? So there are a lot of questions, a lot of details that are required to reproduce a synthetic biology design. And so we wrote this letter to point out the problems, and we started developing this design interchange standard. It was originally called SBOL, but now it's called. It was originally called POBOL, but then a couple of years later it was renamed to SBOL, stand for the Synthetic Biology Open Language. So, what would a design interchange standard be useful for? Well, to begin with, we can exchange DNA level designs with other groups. Now you could argue, well, we already have standards like GenBank, but it turns out GenBank is insufficient for describing the design of a synthetic network. For one thing, you can't exchange incomplete designs. So a group may develop an incomplete design, send it to another group to complete. You can't represent that in GenBank. And also GenBank is a flat file format. It's not hierarchical, whereas we know that designs in synthetic biology tend to be hierarchical, where parts are built on other parts. And you'd like to be able to preserve that hierarchy when you send your design to someone else. The other thing that would benefit from this is if somebody creates something in one lab, sends it to another lab, they make changes and they can send the changes back to the original lab. We can get round tripping of designs without loss of information. Of course, once you have standards, you can start developing repositories. And we saw this with the development of SBML, the Systems Biology Markup Language. The minute that got developed, an entire new ecosystem emerged. And one of the things that emerged were these large databases of models. So some of you may know the Biomodels database in Cambridge, England. That contains hundreds and hundreds of curated computational models. This wouldn't have been easily possible without the development of standard model exchange formats. And the same is actually happening now with the synthetic biology standard. There are now two large repositories that exist now that store designs, parts, etc., and they use SBOL as a means of exchanging designs. The other thing you can do, and was one of the key things that was in my mind, was the ability to reproduce results found in the literature. So recreating a repressolator, for example, is actually not trivial. It's actually quite difficult because there's certain pieces of information missing, and if you don't know that, you can't actually get the repressolator to oscillate. And so one of the key areas I was interested in was being able to reproduce unambiguously what somebody had done in the lab. Now this extends, of course, beyond academia, but to companies. Companies can, once there are standards, companies can start offering catalogs of standardized components. They'll have their own repositories and so on. And we see this also already happening. So, as I mentioned before, SBOL started developing in 2008 with seed funds from Microsoft. We developed the first specification 1.0 in 2010, and last year we released 2.0, which is the latest version we have. And it's now really, although it started with just me and a couple of other guys, it's now developed into a huge community, which I'll mention. And a lot of the work for SBOL 2.0, for example, happened at Utah together with the community. That was Chris Myers. So now we have also proper funding, whereas before, the seed money from Microsoft didn't last very long. We didn't have funding for quite some time, and it was basically run on a voluntary basis. But just over a year ago, we managed to secure some fairly significant funds from the National Science Foundation in the United States to continue funding the development of SBOL. Now, as I said, the SBOL community is now pretty large. These are statistics from June 2015. We've got over 100 people from all over the world that are involved in SBOL, either in its development or using it. We have 30 universities, 14 companies, and 8 other groups, mainly governmental groups. What's interesting about this standard, which was different from the SBML one, was how how much companies were interested. In fact, in the last meeting, half the audience were representative from companies. So there's a lot of interest from the private sector. I should also mention, I mean, we have National Science Foundation funding, but our collaborative group in the UK, in Newcastle, has also significant funding from the Engineering Research Council in the UK. And so what I show here is a map of where many of the developers and users are. Obviously, there's a lot in the United States, interestingly centered on the east and west coasts. A lot of interest in the UK, Spain, and some various other countries. We have two groups in India, which is good. One group in Australia and one or two groups in Asia. So we have a good spread of people across the world. I think this is a standard that's going to stay put and develop. Yeah, you'll see that these actual symbols I'm using here are actually from the SBOL visual standard. So the T is a terminator. This rectangle with a triangle on the end is a coding sequence. This half circle is a ribosome binding site, and this arrow is a operator promoter site. And there are a whole variety of symbols that you can use, and this is recently published in PLOS Biology. So the other thing we have, of course, associated with SBOL is software support. So we have a couple of libraries that have been developed or are being developed that allow you to read and write SBOL data. These are called collectively libSBOL. So currently there's a libSBOL Java library. There's a libSBOL JavaScript library under development, and we're about to release a libSBOL C++ library, which will also have Python bindings. So the idea is you'll be able to manipulate your designs from Python. So as I said, once you have a standard, all sorts of other things start to happen. One thing, of course, is the ability to serialize that data model into other formats, if you wish, and they can be then stored in repositories or converted to other standards. For example, we can convert SBOL into GenBank if you so wish. And we know that the iGEM teams use SBOL, and we also have a connection with the modeling standard SBML, so that now you can associate a model with an SBOL design. So I've mentioned this before, but we'll go over it again. So SBOL can represent a number of things. First of all, it can represent the sequence structure. Importantly, from an engineering point of view, it can represent incomplete designs. So often when you design something, there may be parts of the design which you aren't sure yet about, and so SBOL allows you to put placeholders to say, well, I'm not quite sure what we're going to put there yet, but you know, this, for example, this ribosome binding site has to be in this position, but I haven't yet decided what it is. So you can represent incomplete designs. So this is different from what we normally do in biology, where we describe complete entities. So GenBank describes is a full description of everything. There is no notion really of an incomplete design in biology. I mean, biology is a science enterprise, and it's about discovery. Engineering is about building new things, and so there will be times when your designs are incomplete. So it was very important that SBOL was able to describe incomplete designs. And the other thing we need is some means of describing the function of the various parts and how they connect to form the networks. So we can see in the diagram here, here we basically have the sequence structure. There are certain things here like this ribosome binding site, and this promoter and the terminator is not yet settled. And then we have the network that emerges from the TET-R and the PTET promoter site. The other thing then, which is also extremely important from an engineering point of view, is hierarchical composition. So we can break down a design into multiple modular parts. So for example here, I've shown you this already. We have a detector that's described on the left, and then we have a reporter described on the right, and these two subsystems can be described independently in SBOL, so you maintain their separation. So the idea is, of course, at a later date, somebody will remove the reporter and put something in that's more sort of more interest. So whatever the detector is doing, you actually want it to maybe actuate something else, and so you can, in the SBOL design, you can swap out the GFP reporter and putting something else. So this is the idea of modular design, where you can pull sections out and put new sections in. And the other thing that is crucial to SBOL, so I mentioned before that a lot of information is required in order to reproduce a design. So it's not just the DNA sequence, but we also need things like, what was your strain? What strain did you use? Under what conditions did you grow the cells? How did you measure the output from the design, and so on? And so we have SBOL has the ability to link to other kinds of information, and so basically SBOL becomes the organizing layer that organizes various different kinds of information within a specific synthetic biology design. The other thing we can do is, of course, SBOL can be integrated with other systems. You can either embed SBOL in other data, or you can embed data within SBOL, and you can link SBOL to other data. So rather than embedding the data, you can link SBOL to data or link data to SBOL. And one of the things we're developing this year in my lab is a new extension to SBOL, basically called the host context. And the host context will cover information such as strain information, how the strain was grown, like what temperature was used, when were the measurements taken, what media was used, and so on. So we're creating an extension to SBOL. So SBOL has a natural way of being extended, and one of the first extensions is going to be the host context. So what kinds of scenarios would you use SBOL? I mean, you can imagine, so this image here shows you the things that are involved in synthetic biology engineering. We have obviously users, we have repositories, we have other users, we have automation, right? Things that build and measure designs, and then we have vendors who buy and sell parts. So the simplest scenario is probably sharing. So a user creates a design, drops it into a repository. Another user then retrieves the design from the other repository. So that's one obvious use case. Another one is you're browsing for designs, you're looking for parts, and you want to be able to visualize them. So you would download, you'd browse a repository, which I'll talk about later, and you'll download your parts and your designs, and you'll visualize them to see whether they're suitable or not. You can obviously use this as a design system, so you might retrieve parts from a number of repositories, put them together. You might obtain experimental data also stored in a repository, and you'll merge these together to start the design of a new system. And the second thing, of course, once you have your design, you might want to push it out to either companies who can make the design, or you make it yourself in your own lab using some automated machinery. And this covers the automation of the design. One of the visions, of course, is that all design occurs on the desktop computer. You push that out to a company or an automation that will then just create the DNA, insert it into the host, etc., and what gets sent back to you is then the actually built design. It's a bit like today, where if you're making a PCB board, you'll do your design on the computer, you'll send it out on the web to a company, and a week later you get your PCB boards back. So There are a number of groups now who are setting up repositories and workflow hubs. This one is called the SBOL Hub. It's at Newcastle. It's a repository of generic circuit designs. It's a searchable repository. It has some fairly sophisticated query systems, so you can ask for example, give me all the designs that use GFP, but only in such and such a strain, or give me all promoters that are such and such long, or all promoters that have a certain strength, and so on. And of course, it has the ability to import and export, import SBOL faster in GenBank and export SBOL. So this tool is the perfect tool where you're communicating information between users. ICE is another repository. This is one based in the US. It's funded by the Joint Bioenergy Institute from the Department of Energy. This again is a web service that stores designs. It has some nice visualization capabilities. This is a plasmid. Again, it can export SBOL, GenBank, and FASTA, which you can then use in other tools. This is a visualization service, so if you have pulled a design off one of these repositories, you can then load it into what's called Pigeon, and it'll let you visualize exactly what you have in the design. This is coming out of Boston University, Douglas Densmore's lab. We have additional sophistication in the form of simulation and design. So this is iBiosim from Utah. This lets you design your network on the screen, lets you pick parts to attach to those designs, and then lets you simulate the design as well. And then of course export the design as in the form of SBOL. So this is really a fairly sophisticated tool that you can use to build designs. And what's nice about it is you can modularize designs, swap new designs in and out, and help you develop whatever system you make. This is one of our own we developed some time ago with Tinkercell. We've modified this somewhat recently so that it now exports SBOL, but this is also a visual design based tool mainly focused on simulation and modularity. It has a tab up here which contains all the different kinds of parts that you can use, and you can see this is using the SBOL visual glyphs. We have a fairly sophisticated tool called Biocompiler from BBN Technologies. This is Jake Beal's work. This lets you visualize your SBOL and generate logic designs for gene regulatory circuits. And this is being continuously developed, and it has a very interesting, as you can see, very interesting user interface. This is a very interesting tool from mainly developed at Boston but used at MIT as well, called Cello. This is this is an interesting tool because it uses a standard Verilog format. This is a format used in the electronics industry to specify digital circuits, and you can see this here. So you specify, so if you want to build a gene regulatory network that does some digital operation, you can specify it using this standard format here, and it'll then generate the designs for you based on other information it has on parts and so on, and it can actually create multiple designs at once, so that you can try, obviously a single design you might try, it might not work, but this you can use to generate a whole library of designs, and then through an automated process try out each design in turn until you find one that works in the way you want it to work. So this is this work was published recently, I think, in Science magazine. This is well worth reading what they did. And so I think this tool, for example, points at what's coming in the future, where you have complete automated design of synthetic circuits. Now, one thing I should point out, this is digital, um, and digital is a difficult approach to scale in biology, because each gate has to have its own unique proteins, and being in a liquid environment, it's difficult to avoid interaction. So I'm not convinced that digital circuits will scale easily. I think they'll be great for small things like 5 or 6 gates, maybe a few more. But for large systems, I don't think it would work. I don't think we're going to see any day a digital computer made from a biological system. It just will be impractical. Impractical from the parts point of view. You'd need so many parts, it would just not work. And actually, these systems are really slow. Like these digital circuits can take an hour to switch, so you're not going to exactly get a very fast digital computer. And I think our solid state electronics is so sophisticated, I think it would be a silly idea anyway to create a digital computer from biology. What's more interesting, I think, is the use of analog ideas to make biological circuits. And there was some work republished a year or two ago on the first analog circuits created in E. coli. You need far fewer parts. You can do far more sophisticated computational processing. And if you look at life itself, I mean, most of biology, I think, is analog for various reasons. The other thing to point out is there are various companies that are now supporting SBOL in their tools. Vector NTI is one. I know Genius is another. So we're getting a good take-up from industry. And then another one, J5. This is a new company that's just formed. Has some interesting ideas as well. Again, generating assembly plans. So you have a set of components, and you want the most efficient way to assemble those components into a final design. And also, this group also wants to move all the way to full automation in the laboratory. So if you need to know more about SBOL, we have a whole website dedicated to this, sbolstandard.org. It has lots of information on it, including the documentation, the software. There are community groups that you can join to have discussion and so on. So you'll get all the information you need on SBOL from this website. So, we're always looking for new users and new people. We're particularly interested in experimentalists who might find SBOL useful, but we're also interested in tool makers that use SBOL. But I think at this point it's laboratory users that are most critical, and you need to tell us what tools you would like to have. So finally, just some acknowledgements. Our work at the moment is funded from the National Science Foundation as part of their Bioinformatics Infrastructure Program. Brian Bartley here on the left is the graduate student who's working on the C, C++, and Python bindings libraries for SBOL. They should be released in the next, a beta version anyway, should be out in the next week or two. Brian is also involved in developing the host context extension for SBOL, which will describe strains, etc., for synthetic biology. Deepak Chandran, he was an excellent student, possibly one of the best students I've seen. He developed Tinkercell. That was the design software. Incredibly sharp graduate student. He's now working at Autodesk and doing very well there, I think. Michael Zicki, he helped me launch SBOL, and he was instrumental, in fact, at the beginning for developing SBOL. He's now working for a startup company on metabolic engineering and enzyme design. And then two of my colleagues who are part of the NSF program, John Gennari at UW, who's an informatics person who's extremely useful to have terms of developing the RDF standard and the XML standard and the SBOL standard itself. He's basically providing us with a lot of the computer science. And then Chris Myers, who's an electronic design engineer, who's now a synthetic biologist, basically helped design and develop the SBOL 2.0 standard. He's been instrumental in pushing SBOL forward after Mike went and left and moved on to other things. And of course, we have to acknowledge the SBOL community, which is very large now and growing, and they provide considerable input to the development and where it should go. Okay, I think that finishes my talk, and I want to thank you very much for listening. So I think now we're going to be switching over to Skype, where we can have a discussion on this topic or any other topics you wish to talk about. I'm happy to talk about anything else that comes to mind. Okay, thank you very much. Bye now.